Capillary electrophoresis, or CE, is used for STR typing to separate negatively charged DNA by size using electrophoretic properties. DNA fragments amplified by PCR move through a capillary toward the positively charged anode. CE also separates DNA based on fluorescent dyes incorporated during amplification. Before injection into the CE, DNA samples must undergo sample preparation. First, a master mix is made and added to the CE plate. The master mix includes high-dye formamide and the internal lane standard. A high-dye formamide and internal lane standard are added to each sample. The high-dye formamide is included to ensure that the PCR products are denatured, making them single-stranded. The internal lane standard, labeled with a different dye from the target DNA fragments, co-migrates with the target STR fragments through the capillary. The internal lane standard's fragments of known sizes are used to compare migration times and determine the sizes of the target STR fragments. The dye channels allow for the DNA fragments to separate by color, and they are detected in the order they reach the detection window. When setting up the CE plate, an allelic ladder mix is added every 3 injections. The allelic ladder is an artificial mix of all possible alleles at each of the target STRs and is used to automatically assign fragments by their specific alleles. It is important, though, that the allelic ladder is not added to the wells with our sample's STR fragments, as this would interfere with interpretation by masking or overshadowing the true alleles present in the sample. After the master mix has been added to all the wells and the allelic ladder was added to certain wells, the PCR product for each sample is added to each well. Once all the solutions and samples have been added to the plate, it will be centrifuged and heated to further ensure the DNA is denatured into single-stranded DNA, and then finally snap-cooled to prevent re-annealing of the 2 strands. We want the DNA fragments to be single-stranded as the sizing in capillary electrophoresis is more precise when fragments are single-stranded. This wraps up the sample preparation for capillary electrophoresis, and the samples are now ready to be loaded onto the instrument. Let's watch a simulation of the DNA going through the capillary electrophoresis process now. We can see the wells are filled with our prepared samples. The capillary electrophoresis injects the DNA while applying a charge, which is an electrokinetic injection. Next, the negatively charged DNA fragments enter the capillary, which is filled with a polyacrylamide polymer and has a conductive buffer at both ends. The buffer enables an electric charge across the capillary, causing DNA fragments to migrate from the negatively charged cathode to the positively charged anode. Temperature affects fragment migration speed, so it must be carefully controlled. If too high, the polymer becomes too flexible, reducing resistance and causing DNA fragments to migrate too quickly. If too low, the polymer becomes too stiff, increasing resistance and preventing proper separation. As DNA fragments sieve through the capillary polymer, they separate by size. Smaller fragments reach the detector first, while larger ones pass through last. At the detector, a laser excites the fluorescent dyes on each STR fragment, causing them to fluoresce. This allows fragments of the same size to be distinguished based on their dye color. After data collection, an electropherogram, EPG, is generated by automated genotyping software. As alleles are separated and called, the EPG, also known as a DNA profile, is created. This profile can then be uploaded to databases or compared to suspect samples.